Okay, good morning. So, we will start this session that is about what humans will be after genome editing of human embryonic stem cells. And we got already to the day, in fact, when biotechnology permits us to control and improve the genetic background of living beings and to decide on characteristics of their descendants. Well, so we are opening new doors toward artificially selected, predetermined pathways unknown until now. The rapid emergence of CRISPR gene editing technology intensified the need of evaluation of potential benefits and risks when doing genome editing. So it's now the moment to ask whether it is morally acceptable to modify the human germline using a CRISPR approach. In this session, we will foster the social debate on the use of genome editing technologies on human reproduction, and we will focus on applications altering the human germline that could be pursued, but also in which governance structures shall oversee and control this potential application. We would like to have an interactive discussion with you, and so we will also ask you questions with the system that you probably already saw in the first section. And I would invite my colleague, Veronica Martinez, to come and explain you and give you the codes to enter in the system.
Thank you, Philippa. Thank you, everybody. It's very simple. I think that you are already familiar with the system because you were using it in the plenary session. You just need to take your smartphone, connect it to the Wi-Fi, and then go to the website slido.com. And then once there, you will find a main page where you can type the code of this specific session, which is WSFGE, which is World Science Forum Genome Editing, to make it easier. It is case sensitive. And then if you do that now, at the end of the presentations, we will be launching questions to you to know your opinion. And this is very important that I want to say is that this is anonymous, so you can give really your opinion, knowing that nobody will know what you have answered. And of course, there are no correct or wrong answers. It's a matter of as Philippa said before, to foster the debate about this very hot, sensitive topic.
Okay, so while you are registering in the system, I will speak to you about the panel of people we have here to discuss these questions with us today. And I would start telling that we have Luis Montolio, that is coming from Spain. He works in the National Center for Biotechnology. He's a molecular geneticist, and he likes to say about himself that he is a happy user of CRISPR tools in his lab. So he's a scientist. We have also Ana Veiga from Spain too, and she works in the Center for Regenerative Medicine in Barcelona, and she's an embryologist. So she is in fact the pioneer in vitro fertilization there, and she has a lot of real experience in dealing with the questions of changing human genome. Then we have also Jennifer Mershon, and she comes from Paris, University Panthéon-Assas, and she's a political scientist that specialized in comparative public policy in bioethics issues. She will speak us about how to do the governance of the questions related to changing human genome. And finally, we have Margaret Sliebenfokker, and she comes from UK, University of Sussex, And she specialized in biotechnology and society studies, not only here, but only in Asia. So she has a global view that will be, for sure, very important to our discussion. We will start immediately with the presentations because we have a lot to discuss later, so I would invite Luis to come and start speaking.
Thank you, Filipa and Veronica, and thank you, the Hungarian Academy of Science and the ERC for this kind invitation. So let's talk about CRISPR. This is a revolution. Why it's a revolution? As you know, this has been established for billions of years ago by our friends, the prokaryotes, the bacteria. They learned how to defend from viruses, and now, years later, we're now using this knowledge that we have inherited from the prokaryotes that we are converted into proper genome editing tools. That's why it is a revolution, because it's simple. It is a simple tool, which means that's the power of this tool, and that's the danger of this tool. Everybody can use this tool. This is affordable. It doesn't require investment, doesn't require any sophisticated devices. Basically, it's this yellow patch. It's a nucleases that will cut your DNA or any DNA where this blue molecule, which is the guide molecule, will tell. So basically, we can have a precision. We can really take control of what we do as we hadn't done before. This was in 2012. Now in 2019, everything you see in the screen is commercially available, which means every one of us can purchase these guys and can purchase these nuclease, not only if you are working in a research center, but also if you are at home. We don't have yet a regulation, and anybody of us from home, we can purchase these reagents. So this is what I'm saying, that the use of these CRISPR tools that must come with responsibility. What is the use of these CRISPR tools? You probably have seen this many times, but that's the central part of the history. We are promoting this double-strand break, and this double-strand break that is created by the CRISPR tool has to be repaired, can be repaired the left pathway, and then it will end up with normally the disrupted gene, or can be repaired through the right pathway, which is copying from a template, and then you will end up editing, and you will end up fixing a given sequence in a given place. And that's the power of this technology. What kind of applications can you do with CRISPR? Well, as you can imagine, you can delete a sequence, you can insert a sequence, you can replace a sequence, but also you can do things that you couldn't do before, we simply couldn't do before. And this is what is, I think it's important to highlight. For instance, we can inactivate the nuclease so that the nuclease no longer cuts the DNA. So what's the point of having a scissor that is no longer cutting? Well, the scissor is not cutting, but is placing this particle somewhere in the genome. Then you can associate this with a positive activity, and you can use this tool to reactivate the gene, and this has some phenomenal therapeutical consequences, as we probably have seen these days with the beta-globin gene. You can do the opposite. You can repress specifically a gene, or you can do very imaginative things coming from the brain of David Liu at the Broad, which is associating with other activities, such as base editing, which doesn't require DNA cutting, but just changing an A to a C or a T into a G, or most recently, the prime editing, which is the combination of these different activities that normally they're never together. The evolution hasn't found this way, but now we can basically edit with a precision we didn't have. Of course, that is the future, and there is a variety of applications that we have in front of us, starting from human gene therapy, which is probably what bothers us and what brought us here in Budapest. We need to be aware that not everything is positive. Besides the benefits, we have a number of limitations, and we need to be aware of them. That's the limitations that I express here. The dashed line, above the dashed line, this is the so-called off-target effects. You might be eventually altering genes that are similar, but not the ones that you intended. Or below the dashed line, these are the on-target effects. You are not in control of the repairing machinery, which means that you will be producing a variety of alleles, a variety of genetic alleles, among which there will be the ones that you have intended, plus there will be additional ones that may be even more problematic than the ones that you wanted to fix. So the on-targets is the real problem, and the best way to see is this is one of the mice that we are producing in my lab. I'm using pigmentary genes, so for me it's easy. This mouse should be uniformly black, and it's not uniformly black, it's a mosaic, because not all the cells in this mouse, they have been correctly been restoring the function of this gene. And this is something we need to be aware. And this is something we see, for instance, when we try to transfer a mutation from a patient. This is a patient that we have diagnosed in the lab. She's missing a single letter, and we have created with CRISPR the corresponding animal model for studying, for investigating her disease. And that what she's happening, she's missing one letter. And that's each one of these lines is a different mouse. And the one that is underlined is the one that is missing this C. And this is great. I never had this. But this is 1 in 23. For me, in mice, this is not a problem. I can align the 23 mice, I select the one I'm interested, and I discard the other 22. How do I do this with human patients? So this is the problem. This is the problem. Basically, we have a variety of outcomes when we are applying these CRISPR tools, and this is something to take into account. With animals or with plants, so this is why biotechnology is ahead of biomedicine in these CRISPR tools. We all having, after applying this technology, we all having mosaics, and the mosaics tell us that one of these animals may be carrying the allele of interest. That's the one we select, that's the one we breed out, and we hope that we will be segregating, and eventually we can build the colony and do the experiments. Of course, this meiosis, this breeding, we don't have this possibility with patients. We cannot say to a patient, don't worry, your grandson is going to be fixed. So basically, we have to fix the person, right? So this is the difference between biotechnology and biomedicine. So what about gene therapy? Gene therapy is about fixing a mutation. And of course, we have things that are legal and illegal in most countries, such in our countries. For instance, germline, it is illegal. We are signed the Oviedo, the Asturias Convention, plus they have questionable ethical issues, whereas the somatic gene therapy, whether ex vivo or in vivo, it is legal and acceptable. So we've heard about the potential use of germline. This is the National Academy of Science that it says in 2017 that it is well possible to use germline genome editing, but with a number of conditions, a very precise list of conditions. The Newfields Council of Bioethics, in 1 year later, they went beyond and they said, well, we might even consider doing things for enhancement as long as this is for social justice and solidarity and they are consistent with the welfare of the future of the person. So what happened so far with CRISPR and human embryos, as you will see, most of what we have seen comes from Asia, comes from China. In Europe, we have only one example, the Katiniakan study in the UK. We have one announced from Russia and one published from the US. That's all. And what do we see in vitro? We see very similar things as we see in mice. We see in some cases off targets, in some cases mosaics. So we need to be cautious and we need to remind, but this is what Anna will tell us, that there are alternative methods. So this is the traffic light slide that I wanted to share with you. This is coming from Matt Porteous in Stanford. So the green thing is what we all aim to do. We aim to apply these tools for somatic cell editing or curing. The yellow thing is something that most of the countries it's not possible to do, which is applying this technology for germline editing. Probably is not yet wise, it's not technically acceptable, but it might become in the future. And then what is more doubty, what is more problematic is the red things, which is applying these technologies for enhancement. Well, enhancement is not a science fiction entertainment. This is already a reality. We have very prominent scientists such as George Church that are providing in his lab a list of at least 50 genes whose mutations will provide additional capacities, additional properties to human mankind. So these are physical powers and psychical powers. For instance, if you mutate SNCN9A, you will stop perceiving pain. I don't know whether this is a good thing or a bad thing, that you don't feel pain anymore, or maybe that you are free from diabetes, or maybe that you're free from Alzheimer, or you're free from AIDS virus. So this is the whole list that you can check in his lab. Well, here the problem is that these transhumanists, the people that they believe that the technology should be applied to improve physical and psychical capacities of humans, well, they forget that these genes, they are used for more than one thing. There is a concept that is often forgotten, which is pleiotropy. So we have only 20,000 genes, but we have more than 20,000 functions to carry out. So in activating one gene, it is very naive and very simplistic to say that we will only achieve one thing. There will be a variety of consequences. Of course, then we have in November, just 1 year ago, He Jiankui that crossed a number of red lines, edited human embryos, and then we had these twin girls, and this is being announced this year that this experiment might be repeated in Russia. So these are things that we need to take into account that comes for responsibility. As Philippa said, I'm a happy user. I'm doing things that I could not do before. I remember always that we have to apply these tools with responsibility, and probably we shouldn't be doing all the experiments that we can simply because we can. These are great tools for biotechnology. We will be having CRISPR tools applications in our mouth before it's coming into our blood to test and to treat our diseases. They have been instrumental to investigate many diseases, but we're not in control of the output. There is a level of uncertainty that we shouldn't be forgetting. That's why in vivo using this is probably as today unsafe. Plus, but ex vivo, this is something that we can consider. This is what we have been promoting from this new association that we have created in Europe with an international scope. It's called ARISH. We're promoting responsible genome editing, and this is the Association for Responsible Research and Innovation in Genome Editing. And we basically publish a number of documents, and we can summarize our aims as we shouldn't be using genome editing on human beings until these techniques are deemed safe and effective with precise therapeutic application justified after a broad and open debate. Of course, this is regarding germline editing, regarding somatic genome editing. There is a lot to be said because we have ex vivo and in vivo, and just 2 days ago, as you probably read in the news, we had this first successful experiment carried out from the reactivation of the fetal beta globin copies after repressing the repressor. If we repress the repressor, we promote what is being repressed. And this is the very talented idea behind this new treatment-based ex vivo in hematopoietic stem cells that has been applied by these two companies in the States, and we will be applied also in Europe. And this is to be used for sickle cell anemia and beta thalassemia patients. Well, I finish by saying that this is something that has come to stay. This is, if we put back ourselves in the 80s, there was the PCR, the polymerase chain reaction, that was a revolution. Nowadays, we cannot even conceive a lab without a PCR machine. With the CRISPR, I often say that there's going to be only two types of labs, those that are using CRISPR and those that will use CRISPR. And so I'm thanking my group and all the institutions just providing money, and thank you for your attention.
Okay. Yes, Anna. Thank you. Luis, you respected entirely the time. Congratulations. And now we have Anna. Yes. Yes, this one. Anna, please.
Thank you. It's a pleasure to be here, and also it's a pleasure to follow my good friend Luis Montoliu's presentation. What I will give to you is an outline of what germline genome editing, especially what are the indications to do that, where to apply this, in which kind of cells. I will compare a little bit this methodology with pre-implantation genetic testing, which is the alternative to use this, and I will briefly mention what has been done so far in the human. The first thing we need to focus on is in which kind of cells can we apply germline genome editing. We can use stem cells, and in this case, what we will do is to edit pluripotent stem cells that we will later on differentiate into gametes to give rise to embryos that have been in vitro obtained through gametogenesis. And the idea is to try to use these cells, pluripotent stem cells, that include human embryonic stem cells and also iPS cells, induced pluripotent stem cells. And in that case, the idea is to try to edit those cells that will later on be converted into gametes. In those cases, what we will obtain is a genome-edited sperm or a genome-edited oocytes that can be used further to try to obtain embryos that obviously will be also genome-edited. Other things that we can, other cells that we can also edit are the cells that belong to the testicular niche or to the ovarian niche. In this case, spermatogonial stem cells are the ones that can also be used to make genome editing and to obtain genome-editing sperm. This has been done so far in the mouse, not yet in the human, but this is also a methodology that can be applied to try to edit the germline. Other cells, obviously, oocytes and sperm. In the case of sperm, there is already some experience, not yet practical experience, but in the Stem Cell Institute, an IVF doctor and scientist called Werner Neuhauser says that he plans to use CRISPR-Cas, the gene editing tool, to change the DNA code inside sperm cells. What he wants to do is to reduce the risk of Alzheimer's disease in those kind of sperm, changing a gene called ApoE, and he will be using, this is what he said, this is not a scientific publication, this was published 1 year ago, in fact, and he will be using a new version of CRISPR-Cas technology called base editing. The technology of CRISPR-Cas is evolving very rapidly. I would say that every few weeks there is a new technique that is even better than the previous one. Luis can tell us about this. Now, prime editing seems to be the best solution for certain diseases. In any case, this guy has already mentioned that he will be doing that in sperm. But let's focus on embryos. Embryos are the germline, I would say, leaders, and in this case, editing zygotes or embryos would be the solution to try to modify their genome. We published 2 years ago, 3 years ago now, a paper in Human Reproduction Update that was focusing on genome engineering through CRISPR-Cas9 technology in the human germline and pluripotent stem cells. This was a paper published by the Special Interest Group in Stem Cells of the European Society of Human Reproduction and Embryology, the ESHRE, and it was a review of the critical technical and ethical issues that could deter from employing this methodology in human reproduction, and this was kind of review. If you are interested in reading the paper, the most important highlights are that the uses of genome editing in human reproduction are really limited to a certain number of cases. I will not go through into the detail of this, but talking about genetic diseases, if both members of the couple are carriers of an autosomal recessive disorder, like, for example, Tay-Sachs or like cystic fibrosis, or one member is affected of an autosomal dominant disease, like, for example, familial hypercholesterolemia or von Willebrand disease, I would say that Pre-implantation genetic testing is the method of choice to try to avoid the birth of an affected child. In those cases, we are not modifying anything. We are just selecting the embryos that these couples produce. We are trying to choose, or we choose, the ones that are not affected from the disease. So it's not a genetic modification. This is a very well-established methodology. We are doing that since 1992. In fact, this methodology helps us in vitro fertilization labs to choose the embryos that are not affected by the disease. And in those cases, the safer and easier use of this methodology is the method of choice to try to prevent the birth of affected children. There are reports that say that even pre-implantation genetic testing is the method of choice. We could also use genome editing to try to rescue the embryos that are affected from the disease. And this is a publication from a French group. In fact, it's the Hôpital Antoine Béclair, it's the group of Nelly Friedman, that shows with their results that, in fact, almost more than 900 embryos were affected and were discarded, and in fact, genome editing those embryos could make those embryos available for transfer and in fact increase the pregnancy rate. I agree that this is the case, but I would say that in those couples, the failure rate that is reported in this paper is quite high, and I would say that most pre-implantation genetic testing programs, the results are much better than this one. In other cases, genome editing would be the only solution for certain couples. In the cases of where both members are homozygous for an autosomal recessive disease, in cases in one member is homozygous for an autosomal dominant disease, and in cases, and this is a theoretical approach, one member is affected by a chromosomal structural aberration. What I want to mention is that those cases are extremely rare. It's not something that happens very frequently in couples affected of genetic diseases. So in fact, there is a very limited number and proportion of couples with genetic diseases that could be the good ones to apply this methodology. You have here some heritable diseases that are amenable to gene editing, in this case of the germline. You see cystic fibrosis, Huntington-Myhre syndrome, Light syndrome, and this is a paper that I strongly recommend to the ones that are interested in knowing more about this. This is a paper published this year called Principles of and Strategies for Germline Gene Therapy, and it's a paper of this year. There are a number of limitations, technical limitations, when we apply embryo genome editing. They have already been mentioned by Luis, and I would say that the most important limitations is that only some embryos can be edited, some mosaic embryos can be undetected, and we have the possibility of having errors in the edit that we are performing, both on target and off target effects. And this is something that we need to be aware of. After performing germline genome editing, the embryo has to be analyzed. We have to know if what we wanted to do has been done efficiently. So we have to perform an embryo biopsy and try to analyze this biopsy by doing pre-implantation genetic testing. And you see here that the use of PGD is compulsory, but there are also heavy limitations, because when we analyze an embryo biopsy, what we know is what's in the biopsy, and we need to extrapolate this to the rest of the embryo, which is not always the exact situation. There can be mosaicism that remains undetected, so there are limitations. At the end, sequencing of the full genome should be required to try to detect these off-target effects. And I bring here another paper, a quite recent one, which in fact compares germline genome editing with pre-implantation genetic diagnosis. And the question is there a case in favor of germline intervention? The most important conclusions is that germline genome editing is an attractive option for some parents that do not want to avoid the destruction of embryos. In the case you are doing pre-implantation genetic testing, you discard some embryos. And there are couples, I would say not so many, but there are couples that are not in favor of doing that for moral and ethical reasons. And this methodology, germline genome editing, also offers the possibility to fix affected embryos, as I have mentioned before. As I also mentioned before, pre-implantation genetic testing for monogenic diseases is not always a feasible strategy, and germline genome editing could provide the only possible way the chance to have a healthy biological related children. Let me finish by giving you some information on the papers. Luis has also mentioned that. Most of them come from Asia, from China. They have been published in different journals. I would say that some of them are kind of serious ones and others are more, I would say, not so well-known or not so high-impact journals, but in any case, most of the work has been done first in human embryos that were not usable for human reproduction, in 3PN models, which is wrongly fertilized embryos. Then the 2PN model was adopted. These are normal embryos, and gene correction has been done in a certain number of publications. It has also been mentioned that this methodology can be useful to try to understand the role of certain genes, not only to correct a disease, but also to understand why this gene is important for human development, for example. And as I said, new, I would say, complementary techniques are being put in place, like base editing or prime editing, to try to promote or to improve the results obtained with germline genome editing in this case. As conclusions of my presentation, I would say that genome editing is an excellent tool in the case of the germline for the generation of disease models and to try to understand the role of genes involved in pre- and post-implantation development. I would like to say that genome editing cannot be actually performed with sufficient precision to allow scientists to responsibly contemplate creating genetically modified babies. There are problems related to inaccurate editing and off-target and on-target mutations. But I would also like to say that even if we are not ready yet to use this methodology to produce babies, we should promote research in the human germline. If we want to know if the technique is safe or not, we need to be able to understand what are the problems. And the only way to do that is by promoting research. We have plenty of human embryos that are in liquid nitrogen tanks in the clinics, in the IVF clinics all over the world, that the couples do not want to use for their own reproduction or to donate them, that they are very happy to donate them for research, and I would like to take the opportunity to try to make everybody understand that doing research in the germline is absolutely necessary if we want to reach a conclusion on the fact if this methodology is safe or not and if it's usable in the future or not. So thank you very much for your kind attention.
Thank you, Anna. We've just seen that we have really the methodology, the technology being developed and being used, not yet to in vivo experiments, only some exceptions. But now we should really think about how can we govern this, the use of this wonderful technology that can give us a lot of good outcomes, but that is also still not ready to be used. Now, And so to discuss these questions linked to the governance, we have Jennifer. Can you please? Thank you.
Thank you very much to the organizers for having invited me. This is my first visit to Budapest, and I found it just to be extremely beautiful, and I can't wait to be a tourist part of the time of this conference. This is the outline of my talk. I will first give you a panorama of worldwide regulation concerning germline, human germline cells, and give you contrasting examples between three geographical areas: Europe with the Oviedo Convention, Israel, and Mexico. And then I will present to you different possible governance systems for future regulation, still here speaking about research and or application of human germline editing. And my conclusion will be the suggestion of maybe perhaps imagining the fusion of what is the UK model and the Belgian model. Oops. I want the map. Okay, this is good. This map, okay. This map shows you, and I think it's pretty much up to date, that the regulation of research or on human germline is very contrasted across the world. Pink being a ban on not legislation. Yes, a ban based on legislation, a ban of doing research on germline based on legislation. A faint pink is a ban based on guidelines, so something that is not binding. And the ambiguous gray, meaning that it's probably going on, we don't know how, we don't know why, it's not oversought. Restrictive is light gray, and non-colored countries were excluded in this survey reference. So this is a similar type of map. I think all of the slides will be available to everybody at the end of the conference, so I'm not going to comment each of them. This is another type of slide that speaks to you as to the regulation of human germline editing. Presently in Europe, I give you a few tips and facts. First of all, research in general is regulated at the national level in each European country. And afterwards, for marketing or the sale of a product to be launched, it has to be approved by the European Union. Notably in the realm of what we call ATMP, but I'm not going to get into that. This is just to emphasize to you that Europe, there is the European Union, but each country is going to regulate its own, make its own decision as to whether or not to regulate human germline editing. So a slide on what the ATMPs are. And this brings us to something that is more directly linked to human germline, and that is the Oviedo Convention, and it's a very good example of how Europe isn't really harmonized. You all have heard of it. It prohibits the modification of the human genome for reasons other than health-related, and this is kind of the ambiguous part of the Oviedo Convention, Article 13, for somatic cell therapy that Luis talked about earlier, as well as Anna. There's no problem with that insofar as germline therapy is concerned, or genetic changes in the reproductive cells. Because this can be passed on to future generations, this is not allowed. Now, many countries have signed, including Hungary, France, Spain, because it occurred in Spain, the signing of the convention. Now, this is the sentence that many scientists, researchers are having a problem with nowadays, and there is discussion to transform the wording of the Oviedo Convention. This sentence, an intervention seeking to modify the human genome may only be undertaken for preventive, diagnostic, or therapeutic purposes, and only if its aim is not to introduce any modification in the genome of any descendant. So many scientists are saying that this sentence is contradictory, because on the one hand, yes, you may do so, but it may not be passed on to future generations. Well, if you're going to do so on germline, it is going to be passed on to future generations. So many are arguing that this sentence should be transformed, that another clause should be added onto, except for example, what Anna was talking about earlier, monogenic diseases that are very debilitating or fatal for children who are going to be born with them. So briefly, 35 states have signed it. I won't get into the details. Once you sign it, then your country has to ratify it. Then you can express some reservations and you can have some derogations. What is interesting to see here, for example, is that some countries didn't sign it or ratify it for reasons that were not the same as others. For example, Belgium, which is allowed to create its own embryos for research, did not sign the Oviedo Convention because they thought that it was too restrictive. On the other hand, Germany did not sign the Oviedo Convention because they thought it was not restrictive enough. And finally, the UK didn't sign it because they thought the convention was too restrictive, and as we know, they went ahead with mitochondrial replacement therapy a couple years ago, Kathy Nikian's work that Luis referred to. So now briefly, looking abroad, Mexico is an important destination for therapies that are not available in other countries because the legislation is really difficult to wrap your head around. In general, germline is permissible in Mexico. However, there are regulations that seem to say that it isn't. There is a problem with this because the wording is such that you can interpret it in different ways. And this is where I bring in the necessity to marry together regulation on human germline editing and assisted reproductive technologies, because this is where they're going to be used. This is where couples are going to say, oh, well, if I can go to Mexico and have this germline editing done, it's to have a baby. It's not just for the fun of science or the interest of science. In Israel, we also have a different a regulatory landscape, which is more flexible, but nevertheless forbids cloning and germline gene therapy. However, Israel has an ongoing revision every 2 or 3 years relative to this restriction, and discussions often are pushing for a more relaxed, more flexible approach to germline editing because of disease, serious disease. Now, to almost finish, there are public policy makers that have produced different models that we could imagine to internationally regulate germline, human germline modification. There's the convergence process, which include 3 different types of approaches: regulatory dialogue networking, soft law, or a treaty base, such as the Oviedo. And you have the definition and the examples that follow. There are arguments for and against international regulatory governance, on the other hand. The arguments for, as you can see, would be some sort of harmonization to reduce medical tourism, to prevent the race to the bottom. There are arguments against international regulatory convergence, which speak to the fact that we are all different countries with different political, ethical, cultural differences. International harmonization causes too much confusion, takes too much time, etc. So I would like to advance, because I've been studying a little bit these models and country by country. I'm, of course, obviously not going to read this to you, but I find that I personally find that if we could adapt or adopt some sort of international regulatory model that would emerge the Belgian and the UK model, I think that would be a very interesting initiative. In Belgium, as I said earlier, embryos can be created for research, and it is all very, very strictly overseen and followed and transparent, as is in the UK. Contrary to popular belief, the UK also has a very strict oversight regulatory system and proceeded after a very successful public engagement campaign to pass the mitochondrial transfer law, which allowed this law, and their regulatory instances and organisms are extremely refined, and as well as their public engagement actions, I find. And so Luis mentioned ARIGE. I'm going to mention it too because I'm also a member. This was our launching paper, if you will, and we're very proud of this association because it's international. It's not just Anglo-American like some of the associations or groups we've mentioned before, and we have many members from South America, we have members from Africa, from India, from Middle East. So please join ARIGE. That's my call. Thank you for your attention.
Thank you. So now we have our last speaker of the panel, Margaret. Please. It's down. Yeah.
Thank you for inviting me to speak at this conference. It's wonderful to be here in Budapest, and it's an honor to be able to speak here about social justice, on which I'm going to talk as I was invited to speak about this subject. Speaking about social justice on a topic that is actually going to play out in the future is not easy, so I will be doing a lot of extrapolation. The development of science and technology is not really about ready-made ideas that are worked out in practice. Science is very serendipitous. Unlike standard stories in the history of science, new ideas are imposed onto a materially resistant world, where they are adjusted and transformed. Current choices, we think, can help us decide how to plan ahead, but they are fraught with enormous complexity and unknowns, more so than even the Brexit referendum in the UK. Even when world peace, care, equality and ecology indicate trouble, we still courageously create a wish list with quick fix solutions to our woes. Our wish to have things under control may be blind to reality. The 19th century French sociologist Émile Durkheim saw that in worshipping a totem, we really are worshipping ourselves. Our totem today, I would say, is science, no matter how good it is in many ways. We project our personal health issues onto society, and our totem has become the panacea for our pains. The introduction of regulation, we say, will make sure that the totem is on our side. Most accounts by scientists of how to deal with germline editing view the world as an arena of competing states and conclude that if we do not regulate germline editing, then other rogue competitors will do dangerous things, just like He Jiankui or Denis Vebrikov. This ancient narrative blatantly ignores that the green light for regulating germline editing, no matter how cautious, will be the starting sign for rampant global competition. Genome banks, artificial intelligence, IVF clinics, government regulators, industry and scientists in many countries are in starting positions for the race. The potential fallout from this will be dramatically and literally immeasurable. Human genomic data can provide increasingly dense pictures of health conditions, especially monogenic disorders, but over time, also polygenic conditions will be subject to the enterprise of embryo selection and manipulation. Oh, please. The increased ability to create gametes and embryos on a large scale through IPS and hyperstimulation is likely to lead to its escalation, especially as confidence in the statistical knowledge of the genetic component of features such as IQ and Alzheimer's is increasing. It is questionable whether warnings against off-target gene edits, the omnigenic nature of most genetic diseases, will prevent commercial applications. But as I was asked to discuss the theme of social justice, I will confine myself to issues of discrimination, social values, and competition and experimentation at local, national, and international levels. I will extrapolate what I know of social justice in relation to empirical research into genomics and regenerative medicine and society. The intensification of genetic screening, fetal sequencing, and the growing ability to understand polygenic conditions and traits will increasingly lead us to treat pregnancy as product design. Philosopher Ian Hacking argues that the relation between parent and child will resemble that of designer and designed, entailing new responsibilities of testing and interventions. Attempts to realize our ambitions for offspring will also lead to disappointment and disputes about liabilities and wrongful birth. This kind of liberal eugenics will be experienced normatively as discrimination by those who have less popular traits. Sex selection at present clearly indicates this. And we know from experience with non-invasive predictive testing that parents in rationalistic societies are selective and do not favor children with particular features, such as Down syndrome. Prospective parents will be asked to reflect on what are healthy or better babies or people, for instance in terms of health, physical ability, education and talents. Social pressure exerted through education, state resources, the media and public opinion can lead to the devaluation of certain kinds of people. This will affect the treatment and the identities of other abled peoples, groups with particular features and communities. Thus, when scientist Denis Rebrikov could not find the couples with HIV he needed for embryo editing, he targeted populations with impaired hearing. This flared up heated discussions among the deaf community. Nevertheless, governments and insurance companies are likely to link responsibility for birth to premiums and subsidies. Though some countries have already regulated against this, notably Europe through the GDPR, information societies with genome banks and genome health and testing practices will be tempted to link, for example, employment to genetic makeup. A town in the south of China exemplifies this trend. In Foshan city, successful applicants to civil servant positions were dismissed after genetic testing showed that they were thalassemia carriers. On a national level, we can expect governments' efforts to increase the quality of their populations. In relation to migration, for instance, it is likely that point systems already in use to attract talented professionals from abroad will be complemented by requirements for a personal DNA map. The brain drain from poorer countries will become a genetic drain. In the area of top sports, we see an increasing use of biotechnologies to select potential talent. For example, the company Jiaxuejin in Beijing is already working with the government to sequence the genomes of the national sports team for the Olympics in 2022. And Uzbekistan, as early as 2014, genetically tested children for their athletic potentiality and possible participation in the Olympic national sports team. Embryo selection and germline editing will affect traditional notions of the family, reproduction, human beings, and relations between generations. It will require awareness of genetic identity over generations to come, as genetic change will need to be followed up for reasons of scientific knowledge and health. As germline editing is expected to be experimental for a long time, it will create enormous anxiety and pressure on pregnant women and their families. The use of germline editing will alter the self of both parents and child, as blame, power, and selection will play a major role in identity formation. It will also have far-reaching implications for race, gender, and ability. The increased ability to design and create offspring will weaken notions of gratitude for birth, the acceptance of oneself and one's lot, and the appreciation of unplanned, dare I say, natural diversity within one's community. The desire to be good at sports, education, and so on, apart from genetic propensity, requires efforts and character, a process which forms a large part of life's fulfillment. Rather than relying on accepting children as they come, genetic selection and gene editing risk fundamental conflicts between values of parents and the experience of children. This is likely to increase the existing widespread anxiety, stress, and competition among young people today. Parents, on the other hand, may spend the rest of their lives regretting choices, disappointed with outcomes, disillusioned with errors, and wondering if the newest gene addition or the family genealogy is responsible for their offspring's success or failure. Rather than investing in public health care and health policies that advise people and steer industry, some countries will be tempted to invest in genome banks with the aim to prevent or correct what are regarded as diseases, including Down syndrome, autism, and oral impairment before birth. At present, some countries, like Myanmar, have policies against interethnic marriage, and some leaders advise or require genetic matches before having offspring. Thus, in Cyprus, we find premarital testing for thalassemia, and Ashkenazi Jews test for a range of genetic diseases, while in Iceland, potential mating partners use an app on their mobile phone, informed by the country's genealogy database, to check their genetic compatibility. Governments will be tempted to encourage certain types of offspring. Some countries will encourage couples to reproduce useful kinds of children with, for instance, superior eyesight, cognitive or physical ability to fill a country's vacancies. Singapore in the 1970s encouraged women at university to have children on the presumption that they would have a high IQ. Countries with low health budgets will be tempted to invest in quick fix solutions rather than expensive care. Patients from Nigeria go to Chicago for a CRISPR clinical trial for sickle cell anemia by Vertex Pharmaceuticals. The trial seems to be very promising, but the country would need to improve its clinical and research capacity to take advantage of the technology, which it cannot afford. Some argue that germline editing would provide a more permanent solution. Germline editing is expected to open the door to the instrumentalization of humanity, introducing edits that result in productive, low-maintenance and docile offspring. We can also expect the emergence of policies aimed at what I call gene ethnic cleansing, when offspring are selected and edited based on trends of aesthetics, function, and utility. Edited features will be the subject of political conflict and identity, as ethnicity and cultural values, which is why Voices have already announced the need for a human rights impact assessment. But it is not primarily the rogue states or the evil scientists, who probably do not exist, whose work will lead to ecological mayhem. The development of regenerative medicine clearly shows that it is the initial cautious regulation, such as the EU hospital exemption, that many countries received as a signal that global competition had begun, under the radar or not. As such, regulation is expected and infrastructural preparations are underway. To enable more accurate genetic screening and genome editing, genome banks will aim at storing information on each individual. Countries with less data privacy, such as China, are well ahead in the game. But much research is needed before germline editing can be called a successful practice. According to stem cell scientist Paul Knupfler, Research will use large numbers of embryos and gametes, and its experimental nature will seriously complicate pregnancy and reproductive decision-making. Initially, only the rich may afford safe therapies. High-risk therapy, however, are likely to be accessed mainly by poor and desperate people. If such division is in a country's interest, a genetic class society might become politically acceptable. Though maybe not successful, its politics is likely to be resisted and lead to conflict. If restricted in some countries, international medical tourism is likely to follow. Follow-up care and genetic checkups across generations will be crucial for both medical and scientific reasons, but this may not be feasible as therapy provision and clinical research are likely to take place in grey areas of legality. Just as in the case of regenerative medicine, competition within and between countries will create a rampant industry that will lead to a down spiraling of regulation. Globally, life values, economic investment and knowledge asset will be in conflict. The complexity and uncontrollability of germline editing will create uncertainty and will prompt countries to step up their military security in the form of bioweapons, combining viruses, genetic identity and drones. According to Joe Metzel, a US government advisor, policies on this have been discussed in the US for over a decade in an attempt to ward off disease, bioterrorism and the gene ethnically incompatible. Such security measures are likely to escalate security buildup in other countries. To conclude, the growing opportunity and the encouragement of potential parents to design their families to instrumentalize children will alienate children from their parents, but also those with unpopular traits from the genetically correct community. The entire public is not aware of the strategies of those in power, the corporate aims of data gathering, and implications of germline editing for next generations. One wonders how policymakers decide that merely public debate is all that is needed. When wealthy countries will be deciding in favor of regulation on the basis of some local debate, they will try to set a safe pathway to protect local populations and at considerable cost. But the publics in other countries will not have decided on the same conditions. This makes the poorer parts of populations and medical tourists prime targets for experimentation, and this will have a cascading effect. Thank you.
So, after these 4 presentations that raised much more questions than give answers, we will start by asking you some questions, because it would be also interesting to the discussion to know how do you think about these issues. So we had a small problem. We have to enter again in the system, and you will see the questions. And after, we would like to discuss, so you can also ask questions just after answering our questions in the... Yes, we are almost there. Yes, and I would say also that if we have people following streamlined presentation, you can also vote in answer these questions online, and we will see it here. So we have the first question there. Should there be limits on to human self-creation, given the kind of societies and world we live in? And we have 2 positions to choose between yes and no. Some reasoning there that we can have much more, but we are clearly speaking about the human enhancement through the germline action. And we can see that we have already 9 answers. It's going on, more in the positive. We can start giving some comments here.
So clearly what I can see here is that we acknowledging that we're not in control of what we can do. I mean, it's very naive and simplistic, as I said, that we can apply these techniques without any risk. We're not there yet. And besides the ethical issues associated, first, there's still biological and technical limitations that need to be overcome.
Anna, do you want to say something too? Because you are working in this, in practice.
Thank you. I completely agree with the fact that there are technical issues that need to be solved. One thing that is really important is to clearly differentiate somatic genome editing with germline. And this is something that is very confusing for lay people, for the public. So I think that sessions like this one, what is happening and what are the problems and so on. So it's our duty to do that and to explain what are the differences. As I said during my presentation, I agree that we are not ready yet. There are very few indications, according to my opinion, that could need the use of germline genome editing in embryos. Very limited, but there are. And if the technique is safe, why shouldn't we use it to prevent the birth of an affected child? And on top of that, I have also mentioned in my talk that if we don't do research in human embryos, we don't know. So, I'm sorry not to agree with my colleague Margaret. I mean, there are thousands of human embryos that are staying in tanks, in IVF labs, that will be simply discarded because they are kind of out of the purpose of those couples, that can be used to understand what are the problems with this technique. So I really want to promote the idea of doing research in germline genome editing to understand what are the possibilities of this technique.
Yes, please, Mark.
Because you mentioned this. Yes, well, thank you for making that remark. What I've just been saying has not really been an argument against embryo research. Embryo research is not about individuals. It's not about the individual embryo even. It's about, in this case, scientists going ahead, finding a cure, and trying to understand the embryo. That's what I understand you are doing. And I agree there's much importance to make the strict distinction between somatic and embryonic cells here. But what I've been talking about is based on my observations on the development of regenerative medicine. And what I saw that as soon as regenerative medicine was being regulated in the UK, you could see in China investments going up directly, not in the elite institutes, in all the other institutes, the majority of the institutes. And we see that this is not just in creepy countries. Most of Chinese scientists are just scientists as you, me, and others. We have to take into account it's an international competition. Until now, until 10 years ago, one of the safest and most strict countries in the world regarding embryonic stem cell research and mouse stem cell research was Japan. They had a regulation there that allowed embryo research, but to do anything in practice was nearly impossible. A very, very safe country. Until IPS happened, induced pluripotent stem cells, that allow, for instance, the creation of gametes And IPS was so important, but the fear among the public, among scientists, that the US would be quicker in taking this to the clinic, made the government decide to change the entire law relating to embryonic stem cell research and other forms of embryo research. And so and when this happened, this affected the United States and also Europe. So EMA and the FDA have also been down-regulating their own ideas around what is the best way to go forward. So we're not talking here about individual scientists doing a bad or a good thing, but we're talking about a competition that has, as you well know, created an enormous stem cell tourism around the earth. It's led to billions of dollars going into an area that has achieved relatively little compared to putting billions into the care of people. So we're talking about a different thing, not just about the ethics. We're talking about science, about health budgets, we're talking about competition between countries, and we talk about social justice, and we also talk about ethics. But where the ethics here is in the picture is incredibly hard to define, I find. Yeah.
Okay, Luis wanted also to
Just
say.
very briefly, yes, to Anna, absolutely agree. We need to do research on human embryos. We need to understand what's going on regarding the pre-implantation. But at the moment, no to human germline editing because we're not in control of what's going on. Plus, as a researcher that is working on the rare disease field, I think we have a moral obligation towards millions of patients that are already existing before taking into consideration those human beings that will be born. So for me, it's clear. So we're focusing in the somatic genome editing therapies.
Okay, so we could continue speaking about this question, and we will answer that questions too, but we would put first all the questions there, and we have the next one, and then we come back to all the questions you want to do from the beginning. So now we have another question. If approved, germline genome editing would rather be applied for curing or preventing disease, but not for adding new capacities to healthy embryos. However, if you were to improve traits in an embryo, what capacity would you think would be desirable? And
You can only choose one, yes.
you only can choose one, yes, it's the most one. And we'll get the question we had then there now. You can ask it while we are answering this question. Yeah, please.
The founding father of Western philosophy, Socrates, he was obsessed with words, and he was deconstructing words that people thought were clear. And I think there is a need for some editing also in this hall. It's puzzling that US scientists use the label editing for an activity which is so full of uncertainty. If we think about the context of the word editing, it's related to texts. And we know that we have almost full control when we, for example, are editing a paper. Okay, we might struggle to find new words, new ways of phrasing, but I think it communicates lack of transparency. When you use editing to label this activity, you give the impression, at least to lay people, that this is an activity which is much easier to apply than is the case. So please, I think it is need for some editing. Find a better word.
Okay, Jennifer, please.
Thank you. I really really appreciate that comment, because this, in fact, has been an ongoing conversation ever since scientists or ethics committees have been talking about it. And I have the unique position of being on the French INSERM Ethics Committee, and I also participated in the National Academy of Sciences report that was published in 2017. And I came to the first meetings in Washington with this problematic, because the French were saying manipulation ciblée d'un gène, which means targeted manipulation of a gene, which goes much further than editing. And I was interested in that problematic, and so I went to Washington with that problematic, and it was discussed, but it was determined then that, well, targeted manipulation of a gene is really long, and you know, it doesn't create the image of word processing, you know, cut and paste, which is in fact what these technologies do. So it stuck with editing, and it won't change. It will not go to another word. But I agree with you that this is a very interesting question that linguistics scholars and even anthropologists and other social sciences have already published on this problematic, and will continue to do so as we look back on the history of this technology. Thank you.
Thank you, Jennifer. I would ask now Louise to comment the answers we are getting to the question.
As you can see, if we were at the position that we could provide this additional protection and prevention to cancer, that seems to be the most desirable trait. We're not there, as you know. Well, we know something that happens. For instance, we have elephants. Elephants normally do not develop cancer. And why is that? Probably because they have additional copies of the p53 gene. Well, we could have additional copies of p53 gene, but as you can see, the second option that is mostly favored is it shouldn't be attempted. And I probably myself, I'm in this second position, because we simply do not understand enough of every single gene in order to alter or to operate. We're not there in the case that we are inactivating a gene or we are overexpressing a gene, and we hope that there's going to be only one very clear consequences. There are going to be many consequences. So probably we should go step by step and very carefully.
Any question related to the first or the second one? Well, now we do not have to stay in the question that is being presented. So then we would ask your opinion about the regulation of human genome germline editing, likely to guarantee social and international justice. Do you think that that is possible or not? Yes. Well, Jennifer, you can start speaking about the comments that we...
Well, it depends on what's in the regulation. As we saw with the Oviedo Convention, which is binding, international treaty, if you don't want to be bound by it, then you just won't sign it because you find it too restrictive, which is the case of the UK and Belgium, or you find it not strict enough. So I think it's pretty idealistic, illusionist to think that we will have one day an international binding treaty regulating germline gene editing around the world. Large regional consensus, but I think for the time being, most of this is still going to be carried out on a national level and in coordination with the country's culture and expectations and scientific robustness, etc. So I think it's an illusion to think that we're going to get. The only thing that we could eventually imagine, and that's one of that is called for, is an international ban on germline gene editing, as was attempted with human cloning. But even that wasn't followed by all countries. This happened after Dolly was born. There was this huge international shocked reaction, and most countries then passed laws against cloning.
Yeah, Louise asked also.
So basically, if that would be approved, that wouldn't be as easier and as cheap as probably thought, and that would be very restrictive to those with the capacity to access to these very highly sophisticated technologies, which means to me, in my mind, that will create additional inequalities, which is just the opposite that was originally thought in this Nuffield Council of Bioethics, that we're calling for welfare and fossilized solidarity.
Anna, please.
Yes, what I would like to say is that international regulation is something that we cannot, well, at least I cannot imagine. All countries cannot agree on a regulation. This is impossible. What we should aim is to try to make recommendations that need to be followed. The problem of different legislations in different countries brings us to the problem of tourism from one place to the other to try to reach what you cannot have in your country. So I would say that the aim should be to try to put in place recommendations that should be followed by national academies of sciences and national scientific societies. But at the end, I would say that this competition that was mentioned during the talks is not, according to my opinion, is not bad per se. I mean, it's good that there is competition in the scientific field. It makes scientists to try to improve what they are doing and to have better results. So at the end, I would say that all this progress that we have been seeing is also driven by correct competition among scientists. We all compete among ourselves to be the first in publishing whatever. So at the end, recommendation should be what is needed to try to go all in the same direction.
Okay, Margaret, please.
Thank you. So where I live in the UK, regulation usually means enablement, and it's very important for scientists to have regulation because that's the only way that scientists can start their work. Otherwise, anything they would do, publications or any product patent, would not be recognized. So that's what regulation would be here. When it would start, such in the UK, for instance, then we directly get other countries believing that in future, the future products that will be created by people in the UK or elsewhere will be very valuable. Then they tried to do also research, sometimes very excellent research, in order to get the patents, in order to be able to lead the way in the field. That's very important for science because that makes you a better and also stronger scientist. You get more resources, you get more funding from your government, you get more funding from international organizations and more self-esteem. You can start collaborations with industry, you become known, all good things in themselves. But the competition makes it also that scientists then will collaborate with people in countries that have less strict regulation. So that's where the competition and the international competition becomes a little bit less clean. When in countries, scientists collaborate with other countries because they believe they have a friendly regulation and therefore can attract collaborations from abroad. And this is more is the normal way of doing science internationally. And that's why one has to be very careful with regulation because it easily becomes a tool rather than something that defines how the field should be organized. Thank you.
Okay, so we have another question with, I would say, hot answers because it's not easy to see where do we have. I would start immediately discussing this, reacting to this question. I would ask Jennifer if you want to say something.
I wasn't expecting as many for an immediate international ban, because I was the one who proposed these different. So now it's going up some more. That's quite high. And and nothing on national regulation or governance, whereas that is the reality today. So there seems to be some desire to go beyond the nation state, which has its positive points. But so far, we look like we're still along the lines of something such as the Oviedo Convention on germline. But the Oviedo Convention, as I mentioned earlier, is posing a problem of contradiction to many scientists, and perhaps needs to be revised.
Luis.
So what I'm most worried about, and I think what people fail to realize, is how easy it is nowadays to purchase CRISPR tools. From home, nobody from us, we can purchase radioactivity. We cannot purchase radioactivity, and we understand this. But if you try from home, you will be able to order CRISPR tools, and 2 days later, there will be a courier delivering these CRISPR tools. So the lack of regulation, this is what is enabling things that we probably will have to regret sooner or later. Okay, it's maybe time to go quickly to the last question, because we will have 4 minutes to comment. So we have it there. Concerning the previous question, who should decide? And this is a very important question to our discussion. And we have a lot of possibilities there, scientists, experts, international organisms. But we have also mentioned to regional and national policy makers, and we just saw in the previous question that no one chose regional legislation to solve this question. So it is open already, and let us see. So Jennifer, you would like to comment, no?
Yes, I'm surprised that there aren't more citizens and the public at large, because it indeed is would affect the citizens and the public at large, right? Rather than the organisms or the scientists. Okay, so we got 4%. One of the things that we are very keen on at ARIGE is to bring the public into these discussions, and at a recent meeting, I sort of gave a toolbox of how this can be done, and I'm afraid that if the citizens and public at large aren't more integrated into these kinds of meetings, if there aren't more efforts done to interact with them, then it could backfire. In other words, it could force countries or regions to say, oh God, this is what happened in China is a disaster. We're just going to put a stop to all of this science, which would not be good for science or for citizens in general. So I think previously citizens and the public at large were not involved in ethical debates on what science was doing, we know. And I think it's a good thing that they now be involved in an increased manner, which is not the case today.
Okay, Luis.
So definitely for me, researchers, we are not here to decide anything. So this is not our function. Our function is, our duty, I would say, is to provide information for those that have the responsibility to take decisions. They can take the best decisions according to scientific evidences. To me, the decisions, they should be coming from the elected politicians. That's the one that we elect in democracy. But these people, they should be provided with all the information from all the different stakeholders in order that they integrate all these different sources of information to eventually come up with the best decision.
So, any other comment? If we have any question in the audience, we can still answer one question very quickly, but then it's the end of the session because it's lunchtime now. So thank you very much.